Alright everyone, this will be a short review of mitosis versus meiosis. And it's a little bit confusing, but we'll run through it real quick and make it a little more simpler, hopefully. So starting off with the interphase, it starts with all the 3 growth cycles of the cell before mitosis, or before mitosis ikinesis anyways. Moves on to prophase 1, in which the nuclear end membrane starts to disappear and go away, and you'll start to see the communion cores become more apparent. Going on to metaphase mitosis, you can see through the formation of the metaphasic plate and the lining up of the chromosomes on it. On the anaphase, the chromatids separate, as it says on the bottom, and they go to opposite poles. Whereas when you move on with telophase, the nuclear membrane starts to redevelop again, and you start to see the separation of the 2 daughter cells. Moving on to cytokinesis, separation becomes more clear, and once you get to the actual cytokinesis splitting of the cells, you will have 2 daughter cells. Moving on to meiosis, this is where I'm doing sexual reproduction. Meiosis 1 and 2 are very similar, but have distinct differences as well. So going on to prophase 1, once again you see with the nuclear membrane developing. However, you can tell the difference between prophase 1 and regular because of the amount of DNA. Going on metaphase 1, once again, lining up on the metaphasic plate, preparing to be separated, which they're separated at anaphase 1, and once they're pulled to the opposite core, or the opposite poles, they start telophase. Telophase 1, the nuclear membrane is fully redeveloped with all the chromatids or chromosomes inside of it, and it will start to form a cleavage fur, as we are at telophase 1. Once again, cleavage furrow forms, split the cells. Moving on to meiosis 2, we now have prophase 2, which is essentially the same as prophase 1, except now there's double the amount of information because there's 2 cells. This is not dealing with diploidy or haploidy, FYI. Meiosis 2, chromosomes, once again line up in the center of the metaphasic plate. You can tell meiosis 1 from meiosis 2, or metaphase 1 from metaphase 2, depending on the amount of genetic information. Here's anaphase 2 again, separating at the poles. And lastly, move on to, once again, telophase 2, in which case there are 4 different cells that will be formed afterwards. This is a short little introductory into it. Hopefully you'll learn more. Thank you for watching.